All right. Hello, hello, hello. Welcome back to part two of plants. So, part one was just kind of going over the different tissues and everything and their distributions and kind of where, what is, so that now we can talk about, the xylem and how the xylem works and what the xylem does, and you have an idea of what it is and where it is. So, part two goes more in depth about the vascular tissues, so xylem and phloem. Okay, so first, the xylem, remember, carries water. So it all starts, the root hairs or, the surface area of the root absorbs water via osmosis from a high to low water potential. Hopefully, there's enough water in the soil that there's a higher water potential in the soil outside the roots, so it's able to uptake water. From there, it's going to move across the cortex of the root and eventually get to the xylem vessels that are in the middle of the root. From there, it goes up the stem. It eventually gets to the leaves and the flowers and the fruit and whatever else needs water in the plant. Eventually, it's probably then going to evaporate from the mesophyll in the leaves through those stomatopores, which is going to, we'll see, help drive the concentration gradients that keep the water moving up. So we do know water flows from a high to a low water potential. We've got high water potential in the soil and then in the roots, and the water potential is always going to be lower in the leaves. So water moves down that gradient towards the leaves. Remember, the leaves are a big place, or the main source of photosynthesis and stuff too. So we do need water going to those tissues. The journey to the center of the root. So water, and this is important because it's literally its own objective. So water and solutes. So ions, minerals, certain organic compounds, like other stuff is in the water. So taken up by the root hairs, crosses the cortex to get to eventually the xylem. This is a dicot root. So we've got the blue kind of T shape or X shape xylem in the center via two potential pathways. These pathways are like the favorite, the like blessed child of this chapter are these two pathways. Okay, so apoplast pathway just goes through the cell walls and pretty much around the cells. It never enters the cells themselves. The symplast pathway enters the cytoplasm, enters the vacuole, goes from cell to cell via the plasmodesmata, which remember are the channels between adjacent cells. So if you have two plant cells, they can have, some little tiny channels that let stuff pass from one to the next. So pretty much apoplast is around the cells, symplast is through. So the apoplast pathway, like I said, it enters and moves through the cell wall. So it does come into the cell wall, and so it's in blue here. It does come into the cell wall, but it never breaches that. Never breaches the cell membrane. We're going to call that intercellular spaces. So inter and intra, but inter is between. So the space is between the cells and in the cell walls, but never, ever, 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 ever entering the actual cytoplasm of the cells for the apoplast pathway. The symplast pathway, like we said, is when water does enter the cytoplasm. Water is inside the cells and it's going, across the plasmodesmata. So it's a little bit easier, just goes straight through everything. It's not that one is better than the other or more efficient than the other or favored over the other. It's just it's going to do one of the two things. It's either going to enter the cell or it's not. Okay, so this is where it gets fun. So it travels through the cortex, both pathways, until it gets to the endodermis. Remember the inner cross sections we have. Like that. So endodermis separates the cortex from the vascular bundles. This endodermis has a strip of really waxy waterproof coating. It's called the Casparian strip. The Casparian strip is the structure made of suberin, which is like a thick, waxy, waterproof coating. This stops water from traveling via the apoplast pathway. So you've got these endodermis cells, and this is like a roadblock. for the apoplast pathway. So the apoplast pathway stops before it crosses the endodermis. So this forces the water into the cells if it wants to get to the xylem. So after the endodermis, we only have the symplast pathway. That helps because then all the water is in the same place. It's going to up the pressure a little bit, and it's just going to kind of streamline. Like when you're trying to get into Disney World, and it eventually narrows down into a few lanes to pay the like $130 parking or something. If we were allowed to go places. So after the Casparian strip, after the endodermis, we're only going symplast. We're going to get through the pericycle is just a different type of cell. You don't really need to know that until eventually it gets to the xylem, and then it goes up the stem to the leaves, and we're on our happy way. Okay, so we said xylem transports water around the plant. So because it is just a water tube, you want to minimize resistance to flow. You want those tubes to be as empty and open as possible. So those cells are actually dead at maturity. They, have to be alive at some point to form and grow. But then they die. What's left is kind of like their skeleton made of lignin. Lignin is a really hard, strong, waterproof material. It makes sense that if you're building a tube for water, you want it to be waterproof. So pretty much a mature xylem is just like the dead, empty shell of a cell that once existed. So dead, the xylem's alive. So this is a good comparison. Dead, lots of lignin. So lignified cell walls, strong, dead exoskeletons, just a tube. And it is usually the center of the vascular bundles. Here's a good comparison. So we haven't talked about phloem yet, but the xylem, it is one-way movement. That one way is up. You don't have water going back down to the roots. Water is going up the roots. It's going to evaporate from the leaves, and it's just going to keep going. So water and minerals. There are no end walls between the cells. So like I said, it's the dead, empty shell of a cell that was once alive. So no end cells, cell walls stiffened with lignin. We'll come back to this and talk about the phloem stuff in a few slides. But the phloem is food, sugars, proteins. The phloem is food, sugars, proteins. The cells do have these end walls with these pores that kind of help filter stuff too. And the phloem goes both ways, but we'll get there. Really blurry picture. This is important. Cambridge loves to give these diagrams. Be like, which of the following is the xylem? So the xylem is going to be at the center of the stem, away from the bump of the leaf, away from the bump of the leaf, and the middle of the root. So they could give a diagram like this and be like, which of the following is the xylem? And you'd have to know it. And then all else fails, you're given like a group of cells. Xylem is usually bigger. Okay, so now the phloem. Phloem, food. So sugars, proteins, hormones, pretty much everything that's not water. But there's water in it because it's sap. So pretty much everything else. Vessels are also made of cells. So these are called sieve tube elements. So sieve tube elements stacked on top of each other to give you this, tube. They're separated by sieve plates that have pores in them. These cells are alive because you're dealing with sugars, you're dealing with proteins, you're dealing with things that maybe sometimes need to be actively transported because they're so big, or because maybe we are going against a concentration gradient. So we need to have the ability to spend ATP if we need to. So they do need to be alive, but they lack organelles. So they have really thin peripheral cytoplasms. So they are pretty much empty. They might have like a ribosome or two towards the edge, but they also want to be as empty as possible to let the sap flow as well as possible. So how do they stay alive? With friends. So next to every sieve tube element, there is a companion cell. If I flip back, the companion cell is here. So the companion cell is kind of like this life support system for the sieve elements. Where the sieve elements have almost no organelles, companion cells have a bunch of organelles. So companion cells are making the ATP, making the proteins, making everything that the sieve elements need to stay alive, and is just kind of giving it to them. So it is almost like these cells are on life support, and life support are the companion cells. So they have tons of mitochondria, tons of chloroplasts, tons of ribosomes, tons of everything to make all the products that the sieve elements need to stay alive. So the sieve elements can be mostly empty to allow that sap to travel as easily as possible. Okay, so here's a picture. So we've got, okay, so this is 2 rows. Oh, so parenchyma, remember, is ground tissue. So this is showing, between the ground tissue, we've got 2 rows of sieve elements, and they're all separated by companion cells. So sieve tubes, so this is, 2 sieve tubes, and each sieve tube is made of a bunch of sieve elements, the individual cells, stacked on top of each other. They've got lots of plasmidesmata, those sieve plates with pores, and they're kind of going along. The companion cells are right next door, doing all the stuff to keep them alive. Oh, here's an electron micrograph. So don't worry too much about what branch plasmidesmata means, but we've got plasmidesmata here. You can see the sieve tube member has maybe some stuff around the edges, but not a whole lot. The companion cell, that is a lot of organelles for one cell. And so they're gonna, give stuff to the sieve element to keep it alive. Here's under a light microscope. So you can see the sieve tube members are a little bit bigger. The companion cells are a little bit smaller. Unfortunately, we won't get to use the microscopes for this, but, it is what it is. So like we said, end-to-end to form living tubes. They are alive, they are alive, they are alive. Narrow peripheral cytoplasm. So the cytoplasm is really only like along these very edges. Can have, a few organelles, not a whole lot, nothing crazy, no nucleus. The nucleus breaks down. So really just, the bare essentials, and then the companion cells doing a lot of the work to keep those cells alive. So companion cells are modified parenchyma. So remember, that's ground tissue. So they're just a specialized type of cell. And again, their job is to keep the sieve elements alive. They are metabolically active, way more metabolically active than a lot of other types of cells. Really dense cytoplasm, small vacuoles to make room for extra organelles to keep, because they're doing enough work to support 2 cells pretty much. Synthesis. Oh man, okay. So that is the end of part 2. We're splitting this into 3 parts because as a whole, it's a big chapter. So end of part 2,